Hi, everybody. Welcome to today's Hot Topic program, Uncovering Our Past Using Genomes to Understand Human Evolutionary History with Dr. Priya Moorjani. My name is Brianna Pobener, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. I'm a brown and gray haired woman in a blue shirt wearing headphones in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us or you've attended before, we're so glad to have you here. Before we get started, I have a few housekeeping notes. This discussion offers closed captioning. You can turn the captions on or off via the CC button, which should be located at the bottom of your Zoom interface. We're in a webinar format, so we can't see or hear you. As you have questions, please go ahead and submit them to the Q&A box, which is at the top or bottom of your screen. It looks like two speech bubbles, so that we can sort through as many questions as possible. The Q&A time really flies by. The Q&A box is also where we will share any relevant links during the program, so keep an eye out there. We'll start with a presentation by our speaker, Priya Moorjani, and then I'll join her here to take your questions. During the presentation, I will also write answers to some of your questions, at least any that I can answer, as will another member of our behind-the-scenes team, Dr. Ryan McCrae. So now I'd like to go ahead and introduce our speaker. Dr. Priya Moorjani is an assistant professor in the Department of Molecular and Cell Biology and the Center for Computational Biology at the University of California, Berkeley. Her lab uses genomic data from ancient and present day individuals to study questions in human genetics and evolutionary biology. To this end, they develop computational methods to understand three things. One is how different evolutionary processes, such as mutation rates, evolve across primates. The second is when key events, such as introgression and adaptations, occurred in human history. And the third is how we can leverage these patterns to identify genetic variants related to human adaptation and disease. And now I'm excited to hand over to Priya.
Thank you, Brianna, for the introduction. I'll go ahead and share my slides.
Great.
Thank you all for joining today. I'm delighted to share our lab's research on studying genomes to understand our evolutionary history. Our genomes contain a record of our ancestors. In each generation, we inherit about 50% of our DNA from the previous generation. So each of us has 50% of our DNA from our parents, 25% on average of DNA from our grandparents, and so on in every generation. So if we zoom into our genome, here I'm showing you the genome of one individual, and I'm showing you the 23 chromosomes, and different bits of our DNA come from different ancestors. So our genomes are really a mosaic of little bits of DNA from each of our ancestors. Here, this bit comes from this ancestor in the distinct past, and another bit comes from an ancestor more recently related to us. And this is the kind of information that we use in our labs to learn about our evolutionary history. To tell you more about it, we use genetic data that we get from living people, as well as data of people who lived in the past, people like Neanderthals who are no longer extinct, but because of very sophisticated sequencing technologies today, we can actually get DNA of all of these individuals and learn about how they relate to us. And from studies of present-day data, we know that population mixture is recurrent throughout human history. Now, this term population mixture, admixture, or integration is something that I'll use interchangeably. And what I mean by that is that populations that were previously separated from each other, often we find in history mix with each other. And these processes are very common. We find evidence for this at very recent timescales when we study genomes of of, say, African Americans, Latine populations, where the mixtures are very recent, over hundreds, in the past hundreds of years. But we also find evidence of these mixtures thousands of years ago in Europeans and South Asians. And all non-Africans have evidence of gene flow from Neanderthals. Now, these are just a few examples that I chose. In fact, this process is so common that we find evidence in almost every individual's genomes, because many of our ancestors were separated at different times in history, but when they come together, there is mixture between these populations. And today, for the rest of the talk, I'm actually going to focus on this first mixture. I'm going to focus on this first mixture, which is the gene flow between Neanderthals and modern humans, for which Svante Pääbo won the Nobel Prize in 2022. Now, as many of you may know, modern humans arose in Africa about 200 to 400,000 years ago. And then there was a migration of people out of Africa, which occurred about 40 to 60,000 years ago. But when we came out of Africa, we were not alone. There were other groups of humans living there, like Neanderthals. And when we met with Neanderthals, there was mixture between our ancestors and Neanderthals, such that all non-Africans today have about 1 to 3% of Neanderthal ancestry. And we also find that there was mixture between other groups that were also living outside of Africa. From the sequencing of a pinky bone that was found in a cave in Denisova, which is in the Altai Mountains, we now know about a distinct cousin of ours, the Denisovan. Here I'm showing you two pieces of, two fossils of Denisovans, one, the little pinky bone that was sequenced, and then some other teeth that were found in the same cave. And we know from studying the genomes of present-day people that Denisovans also interbred with modern humans. And today, most Oceanians and Asians have some amount of Denisovan ancestry. In this case, about 0.1 to 5% of Denisovan ancestry. And when these diverse groups mix with each other at a genomic level, what we find is that in each generation, we get one chromosome from our parents. So here, this individual has one chromosome from its Neanderthal parent and one chromosome from the Denisovan, one chromosome from the modern human parent. Now, here I'm coloring their genomes based on their ancestry. Neanderthals and modern humans were separated for about 0.5 million years. And so there are many differences in their genome, so we can do what we call chromosome painting and color the genome based on their ancestry. And then the first generation would get one chromosome from each parent. But what happens in the next few generations is that in the formation of egg and sperm, there are crossovers that occur between our parental genomes. And so now there would be mixing of these two genomes, and then there would be a bit of Neanderthal ancestry on the modern human chromosome and a bit of bit of Neanderthal ancestry on the modern human chromosome, and a bit of the modern human ancestry in Neanderthal chromosomes. And there would be, this process would repeat in every generation, and then there would be more and more crossovers, such as these bits of Neanderthal ancestry become smaller and smaller. So many generations later, if you look at present-day people living today and color their genome by ancestry, what you find is majority of our genome has modern human ancestry, similar to what Africans have. But in some bits of DNA of non-Africans, the DNA matches better to Neanderthals. So there are these little bits of DNA. Because this process of crossovers occurs rather randomly, what you find is if you take two non-African individuals, they might have Neanderthal ancestry in different positions of their genome. And this process continues in every generation, and you find as more and more individuals are sequenced, we can recover more and more Neanderthal ancestry. Now, let's think of how much Neanderthal or Denisovan ancestry is present in modern humans. Here, I'm showing you first data for Indians. As you may have guessed from my name, I'm from India. And if we look at different individuals living in India on this plot, each line is individual. And on the y-axis, I'm showing you the proportion of the Neanderthal ancestry you recover from the living individual's genome and the Denisovan ancestry that you recover. So on average, Indians have about 1.5% of Neanderthal ancestry, and they have about 0.2% of Denisovan ancestry. Here I'm showing you data for 2,500 individuals, and mostly you find that most of the individuals have similar amounts of Neanderthal and Denisovan ancestry. But if you go go and look at data from other people living in, say, East Asia, Europe, Middle East, or Oceania, you find that the picture is more interesting. People living in East Asia actually have about 20% higher Neanderthal ancestry. People living in Europe have a little bit less Neanderthal ancestry. The picture is even more interesting when you look at Denisovan ancestry, and in this case, people who live in Oceania, here shown in green, have lots of Denisovan ancestry, about 2.5% in this case. And then when you look at other people living around the world, you find that like South Asians, East Asians also have about 0.2% Denisovan ancestry, but people living in Europe have very little Denisovan ancestry. Now, as I mentioned earlier, that if you look at different individuals, they have Neanderthal and Denisovan ancestry in different places in their genome. This means that actually that we could take these little bits of DNA from different people living and try to reconstruct the Neanderthal ancestry just by looking at DNA of modern people. And this is what we tried to do in a study. And what we find is on the bottom, I'm showing you Neanderthal ancestry, and on the top, I'm showing you Denisovan ancestry. And here, as we increase the number of people, we can actually reconstruct more and more of the Neanderthal and Denisovan genomes. So what you find is that if you sequence, say, about 3,500 people living in South Asia, you can reconstruct almost 1.6 gigs of the Neanderthal genome and about 0.6 gigs of the Denisovan genome. That's about half of the Neanderthal genome and quarter of the Denisovan genome. Now, if you look at the direct sequencing of the Neanderthal genome, that's also about the same size, just because often ancient DNA data that we can recover is a little more damaged, because as time passes, the DNA gets degraded. And when we sequence that, we have to apply careful filters so that we know that we are really looking at authentic sequence. And so rather than recovering the full genome of Neanderthals, after filtering, we recover only half of their genome. And so similarly, in this case, we are actually now able to recover data of Neanderthals from our own genomes, which is about the same size. Now, you might be wondering, what is the function of these bits of DNA that we have inherited from our Neanderthal or Denisovan ancestors? And to answer this question, what we often do is we look at places in the genome where there is a lot of modern humans have retained Neanderthal ancestry. So here I'm showing you each line is a chromosome, and on top I'm showing you the distribution of the Denisovan ancestry, and the bottom in blue is the distribution of the Neanderthal ancestry. In some places you find that the colors are really dark, and these are the regions where many people living today have Neanderthal ancestry. If you zoom into one of these positions, what you find is that a major risk factor for COVID-19 was inherited from Neanderthals. And this risk factor, this is a position on chromosome 3, and variants in this region would increase the risk of hospitalization post-COVID. But you can also look at other places in the genome. And in this case, we find sometimes Neanderthal ancestry was actually protective for COVID. You also find that on typically many positions in the genome where we have Neanderthal ancestry are related to adaptations that we environmental adaptations that you see outside of Africa. Some are related to skin pigmentation, others are related to immune function. This might be because Neanderthals were adapted to these environments, and so when we mixed with them, we inherited variants that helped us adapt to these environments. And one of the foundational examples in this field is of a bit of DNA that Tibetans have inherited from Denisovans that have allowed them to adapt to high altitude living. Now, not all positions in our genome have retained Neanderthal or Denisovan ancestry. There are large regions of our genome that are completely devoid of archaic ancestry. I will use this term archaic to often refer to Neanderthals and Denisovans in a similar way. And we use this term because by archaic, we mean separation very long time ago. As I mentioned, we separated from Neanderthals and Denisovans about 0.5 million years ago. And so we call these splits to be very old or archaic. If we look at some regions in our genome, we find that there is nobody who has Neanderthal or Denisovan ancestry. These regions tend to be very large, and in fact, there are 5 that in many studies we have seen where no living people has Neanderthal or Denisovan ancestry. And this might be because Neanderthals and Denisovans were living in very harsh environments and tended to have very small populations. And so they might have a lot of deleterious mutations that when they interbred with us may have been selected out. And I'll come back to this point in a few slides again. Now, you can also look at the archaeological record to study how far were these groups spread. Now, for Denisovans, as I mentioned, we only have very few fossil evidence from this one cave, the Denisova cave. Slowly, as we have learned about this group, we are now starting to collect more samples, and we are finding that maybe there is a few samples in outside of Denisova cave in the Tibetan plateau where Denisovans are also found. But mostly what we know about them is actually from their DNA in our genomes. But for Neanderthals, we have a very rich fossil record, and work by Tom Hyam and Katrina Duca and many others have helped us understand how far they were spread and when they might have gone extinct. So in a study, they collected data for 200 samples from 40 different sites across Europe and West Asia, and they tried to infer how long Denisovans might have been present at these sites. So in the bottom of this plot, you can see that there are these little distributions, and these distributions are of the presence of Neanderthals in different regions, and the large uncertainty is about accounting for the timing estimates of different individuals. Based on this, they have suggested that maybe Neanderthals were around up to about 39,000 years. Similarly, looking at data of modern humans, we can also study when modern humans may have been present at the same sites overlapping with Neanderthals. And based on this, they've inferred that modern humans and Neanderthals may have overlapped for about 6,000 to 7,000 years. Now, you might be wondering, when did the gene flow between modern humans and Neanderthals occur? Possibly around this time, but was there one pulse of mixture in all common ancestors of non-Africans? Or given that East Asians have a little more Neanderthal ancestry, maybe there were secondary pulses in those groups? And this is the question we tried to answer by looking at ancient DNA data. And we use the idea as geneticists to use genetic clocks to infer the timing. Now, I showed you this plot earlier where I told you how population mixtures lead to these mosaic chromosomes. But as you can see that if we are soon after the gene flow, there are big blocks of Neanderthal ancestry in this individual's genome, and many, many generations later, these blocks are become smaller. Now we could use this insight to try to infer when the mixture occurred. If we look at genomes and we find very short blocks, we know that the mixtures must have occurred a long time ago. And if we find very long blocks, we know that the mixture must have occurred recently. And so this way we can use this we can use the block sizes as a clock to infer when the mixture occurred. We can also look at present day as well as ancient DNA sample. I mentioned that we now have the ability to sequence our ancestors and study those. Now, if the mixture occurred, say, around 50,000 years ago, as we saw from the previous slide, where modern humans and Neanderthals overlap with each other, and if we look at present day individuals, a lot of time has passed since then. And so the blocks that we see in present day living people is very small, tiny little blocks. However, we could actually go back in time because ancient DNA allows us to time travel and look at people who lived in the past. And in this case, we would say if we had a 40,000-year-old sample or 45,000-year-old sample like this one, Ustashim, then we would expect very large blocks in this individual's genome. So this is the kind of data that we tried to use to learn about the timing. In a recent study, we looked at data from 58 modern human 58 ancient DNA samples. We tried to also use samples of individuals that were sampled a long time ago. These are individuals like Oasa that lived 40,000 years ago, or Bachakiro that might have lived 44,000 years ago, or Ustashim that I showed you on the previous slide that lived 45,000 years ago. And then we tried to identify where Neanderthal ancestry is in their genomes to study in further timing. Now, let me show you what I mean by that. Here is data for one individual called Malta. This is a 24,000-year-old sample that was found in Siberia. Here is the 23 chromosomes of Malta's genome, and you can go in and identify bits of DNA that match Neanderthals more than they match modern humans or African populations. And these are shown here in the red lines. You can see that these are very tiny bits, but we can actually measure the lengths of these bits, and then we can infer that the gene flow in Malta occurred about 23,000 years before Malta lived. Now, because it's an ancient DNA sample, any kind of biological clocks would stop when the individual would die. And so if you need to know the time before present, then you have to add the age of Malta back. And in this case, we inferred that the gene flow occurred 47,000 years before present. And we can also look at another genome, in this case, Oasa. Oasa is a really interesting sample that was found in Romania. When there was a lot of debate about whether Neanderthals and modern humans mixed with each other, especially before the sequencing of the genome, there were very few samples which had morphological traits that were intermediate to modern humans and Neanderthals. But Oasa is one outlier which actually has morphological traits that are intermediate to modern humans and Neanderthals, such that if you look at Oasa's femurs, you think it might be a Neanderthal. And if you look at Oasa's skull shape, then you might think he's a modern human. Now, if we look at Oasa's genome, you can see that there are very large blocks of Neanderthal ancestry in Oasa's genome. That's because in case of Oasa, the gene flow occurred 200 years before the sample lived, which is only about 6 generations, which means that Oasa is a great-great-grandchild of a human Neanderthal mixture. It's samples like Oasa today that have pressed the debate about whether there was gene flow between modern humans and Neanderthals, because we can see through these ancestry paintings that there are large blocks of Neanderthal ancestry in our ancestors. We can do this analysis with many more samples, as I showed you on the previous slide, that we have lots of ancient DNA samples. And so we applied this method to about 25 different samples here. And we inferred that the mean timing of the Neanderthal gene flow was about 47,000 years before present. But we can actually jointly analyze the data for all of these samples and not just learn about the mean time of the gene flow, but also the duration of the gene flow. And when we do that, we infer that the duration was about 6,800 years. This means that the most likely date for the Neanderthal gene flow was between 43,500 and 50,500. And this very closely overlaps the evidence that we had seen from the archaeological material. Now we can also start looking at the functional variation to try to study if these variants that are present at high frequency in present day living people, is this because of selection, or is this because of just drift? If it's because of selection, what were the selection pressures? And in that case, learning about the timing can be quite helpful. So that's what we tried to do. We tried to look at these variants, but we tried to look at when these variants arose and what was their frequency over time. So if we look at one of these variants, this is a variant that in a gene called BCN2, which is related to skin pigmentation. If we look at samples that are 45 to 30,000 years, what we find is that this variant is present in slightly low frequency. And then as time increases, we find 30 or 10,000 years ago, its frequency increases, and 10 to 1,000 years ago, it's even higher. And today in modern humans or living people, it's actually quite high frequency. We can also do the same for other variants. If we look at another gene, in this case, the variant was at low frequency, but not until 10,000 years, it did not really increase in frequency, but then something changed and it quickly increased in frequency in present and is at high frequency in present day individuals. And this might be a variant that has recently become adaptive. Maybe we inherited the raw material from Neanderthals, but in that environment, it was not particularly that important because it didn't provide any, say, major adaptation to the environmental conditions. But as the climate changed, maybe as things changed after the last glacial maximum, something changed in our environment that made these variants be beneficial. Now, remember, I also told you that some regions of our genome have no Neanderthal or Denisovan ancestry, and we often refer to these regions as archaic deserts. Now we can actually go back in time and see how these deserts were formed. Here, I'm showing you results for one of the deserts. This is a region on chromosome 7. It's a large region where Neanderthal ancestry is depleted for more than 1 megabase. And then I'm showing you all the ancient DNA samples, but this time I have organized them by time. And we see that in 45,000-year-old samples, you don't find any Neanderthal ancestry in this region. You go down the line and there is very, very little Neanderthal ancestry, if at all. And even when you do find some variants are shared, they are very tiny little blocks. Only when you come out to outside of this region, you start finding some amount of shared Neanderthal ancestry. We can do this exercise for other deserts also. And we find the same pattern, and very rarely do we see any Neanderthal ancestry. Maybe in this last desert, we find some Neanderthal ancestry in a few individuals. And what this is telling us is that these deserts were formed very quickly. Maybe Neanderthals had some deleterious variants that would not allow the people who had these variants to survive. And so they quickly got purged from modern human genomes. And then what was left behind is possibly mostly neutral. So this might suggest that maybe there was much higher amount of Neanderthal ancestry when we first inbred with Neanderthals. But today there is less Neanderthal ancestry left. And we can also look at that question by looking at our sample stratified by time, and then see how much Neanderthal ancestry is present in genes, which might be at high constraint compared to regions far away from Neanderthal ancestry. Far away from genes which might not be constrained or might not have as many functional variants. And so if we look at present day people, what you find is that that's about the same. We have about 1.5% Neanderthal ancestry in regions outside of genes. This is the genome-wide average estimate I told you about. But if you look in genes, there is actually less because some of the functional variants that were deleterious may have been purged. But if you look at ancient DNA samples, especially close to the gene flow event, what you find is that initially there was about 2.5%, there was about 2.5% ancestry in our coding regions. But if you go away from these constrained regions, what you find is there was much higher, almost 5% of Neanderthal ancestry. This might suggest that the initial mixture was much higher, more than 5%, and today what is left is just 1.5%. Now, just to summarize our findings, by using data from living and ancient DNA samples, we are able to reconstruct a more deeper picture about our mixtures with Neanderthals. We find that there is evidence for a single extended period of gene flow with Neanderthals, overlapping between 43,500 to 50,500 years ago. This is nicely consistent with the archaeological evidence. We can also study patterns in these early modern human samples, and we find that many of these groups were very close to the Neanderthal gene flow event and have large bits of Neanderthal ancestry. And natural selection, both positive as well as negative selection in the formation of the deserts, occurred very rapidly after the gene flow, and we find evidence for that in our very early modern human samples also. However, admixture or gene flow is a recurrent theme in our history. We have just started to learn about this from looking at a few examples, but actually our history is full of these mixture events where you see that there has been gene flow between modern humans and Neanderthals and Denisovans, but there's also evidence of gene flow between Neanderthals and Denisovans, and there's also possibly some evidence of deep lineage in Denisovans that through Denisovans may also be present in our genome. Based on the split time, some people think that this might have been footprints of Homo erectus ancestry, and we might, by studying present-day people, be able to even learn about that ancestry. And Africans are, while they might not have a lot of Neanderthal ancestry, they also have small bits of Neanderthal ancestry. And this is not because of direct gene flow from Neanderthals into Africans, but actually because of gene flow from non-Africans into Africans, and that which brought Neanderthal ancestry into their genomes also. Africans may also have other archaic ancestries, given that we find that our fossil record is so rich in Africa, and there were many different groups of humans living there that people may have mixed with each other in Africa also. Now, with that, I would like to thank my collaborators, in particular, Manjusha Chintalapati, who's a postdoc in my lab, and Leonardo Ayasi, who is a PhD student at Max Planck Institute. The project I told you about of Neanderthal ancestry over time was done in collaboration with Max Planck Institute with Ben Peters' group in Max Planck. I also shared how we can learn about archaic ancestry through modern humans, and that was work led by two postdocs in my group, Elise Kurdenkopf and Lauritz Koff. And I would also like to thank my lab for critical feedback and suggestions throughout the projects, and our funding sources for supporting this work. And thank you all for joining today. I'm happy to take any questions.
Thank you, Priya. That was fantastic. And we already have a bunch of questions that have come in, so I'm just going to launch right into it. And I'm going to start with this one because you alluded to this a little bit at the end, this question from Niels. Could the analysis of modern human genomes suggest the possibility of otherwise unknown extinct hominins?
Yeah, there is actually a lot of active work in this area, because we can look at modern human genomes and try to study which regions look like other parts of the genome or which look very unusual. Sometimes you can find a region that has a lot of footprints of deep lineages. There might be a lot of mutations in a particular region that are uniquely present in some modern humans versus other modern humans. And that kind of information can help us infer that maybe there was mixing with other unknown archaics, which we don't have genomes for.
Thank you. This question came in actually early and also just now. So John asked, are there European or American individuals with no Neanderthal or Denisovan DNA? And Ben asked, do any people not have any Neanderthal or Denisovan DNA?
Yeah, so there is a lot of evidence that the gene flow occurred in the common ancestor, Neanderthal gene flow occurred in the common ancestor of all non-Africans. And so far, I think studies have included thousands of samples, and there is evidence for 1 to 2% ancestry in all non-Africans. Sometimes you might find that there is no DNA, but often it's turned out that it's because of the markers that you're looking at in the genome. Sometimes if you're not looking at the full genome or the quality of the data is not very good, then you might find that maybe there isn't enough evidence for the gene flow. Africans, as I mentioned, also have evidence for Neanderthal ancestry because of back gene flow from Europeans. And so there seems to be pretty consistent evidence that most non-Africans in particular have Neanderthal ancestry. When it comes to Denisovan gene flow, the picture is a little bit different. I think mostly people in Oceania and Asia have evidence for Denisovan ancestry. Even people in Asia have only 0.1% of Denisovan ancestry, so it's very easy to miss. Although our genome is 3 billion bases, so 0.1% is still quite big. But it slowly starts getting at the detection thresholds, and so sometimes you can even miss that.
I'm going to jump to this question because it sort of follows on what you were just talking about. Laura asked, are Australian people part of this study? Do they have higher amounts of Denisovan DNA like people in Oceania?
Yeah, that's absolutely true. Australians have higher amounts of Denisovan ancestry. They were also part of the study. And in fact, there are others also who have looked at data from Australians. And you find clear evidence of Denisovan ancestry almost as high as Oceanians. And it seems that the gene flow may have occurred in the common ancestors.
Oh, thank you. Here's a different question from Lynn, who asks about how many Neanderthal and Denisovan genomes have been sequenced to date, and are they whole genomes that have been sequenced with long reads, such as nanopore sequencing methods?
Yeah, so so currently there are 4 high coverage archaic genomes, 3 Neanderthals and 1 Denisovan. And these are very high quality genomes. They have been covered to a depth of 30 or 40, which just means that every position in the genome was covered 40 times so that you can very confidently infer the sequence. There are also a few more low coverage genomes for both Neanderthals and Denisovans, though sometimes they tend to be have very limited information and are often not always used in these analyses. There was another question I missed.
Hold on, let me look back. Actually, hold on a second. It was, are they whole genomes that have been sequenced with long read methods?
Yeah, so when it comes to long read methods, actually people have tried with PacBio. I don't know if anyone's currently tried with Nanopore, but one challenge with ancient DNA data is that as time passes, the DNA becomes very fragmented. And so actually the long reads are not particularly beneficial in this case because the DNA is already sheared up. And so unless the DNA is very well preserved, doing long read sequencing in these cases is not very helpful.
That makes sense. Thank you.
Yeah.
Here's a question from Eileen who asks, is it possible that Neanderthals and Denisovan have fewer chromosomes than modern humans?
No, we know from studying their genomes that they have the same number of chromosomes as modern humans. They only separated from modern humans about 0.5 million years ago. And so for reference, humans and chimpanzees separated about 8 to 10 million years ago. And so this is only a tenth of the time. We know that chimpanzees and modern humans differ in their number of chromosomes because of a fusion that happened on chromosome 2, but Neanderthals and Denisovans share that with us.
Thank you. Jason asks, do we see any kind of sex-biased admixture between archaic individuals?
Yeah, there is. So the typical way we study sex bias admixture is by looking at either mitochondria or Y chromosome and try to
Mhm.
compare how much ancestry is present in those chromosomes compared to what you see on the autosomes. And when you look at mitochondria, we don't have, humans don't have. We have the modern human mitochondria. We don't have the Neanderthal or Denisovan mitochondria. And similarly, when you look at the Y chromosome, we don't have the Y chromosome from Neanderthals. And so there could be sex bias, but what we think is there is also previous gene flow events. And now we know that Neanderthals actually have the modern human Y chromosome. And so it's been a little bit hard to tease apart the sex bias.
Thank you. Here's a question that kind of follows on that one nicely from Aurelia and Sebastian, who ask, how long ago did humans and Neanderthals start breeding together?
Yeah, we think that about 51,000 years ago, they might have started, they might have interbred with each other. This is also consistent with the fossil evidence. And if you look at data from present day or ancient DNA samples, a similar estimate emerges.
Excellent. Thank you. So Carl and Urate ask, could you comment on the observed desert in Neanderthal ancestry in the male reproductive genome area? What might have caused this?
Yeah, it's actually been a kind of a mystery what it might have been because typically you find evidence for such deserts because of reproduction isolation. When two groups are becoming a species, you find that they might have mutations on separately on their lineages, such that when those mutations come together, you find some incompatibilities. Now, that was previously the thought that this might be the case. But actually, the fact that that all of us are living here today with Neanderthal ancestry in our genomes suggests that they were not that different. And so that we were not really species to have such differences. It also takes millions of years to form different species. And so 500,000 years of separation is actually a very short period of time. So more likely, explanation could be that Neanderthals just had a lot of bad mutations or deleterious mutations causing diseases, which in their small populations they were not able to get rid of. But when the mixture occurred, our DNA have higher fitness, and so it got selected, and that's why we see these large regions without any Neanderthal ancestry.
That makes sense. Thank you. And
Yeah.
actually, this question follows on that very nicely. So Ron asks, could you discuss how the Neanderthal genes modern humans have today must have been beneficial or part of haploid groups that are beneficial? Otherwise, Neanderthal genes would have been purged by now.
Yeah, some of them also are beneficial. We find that, um, conversely, also modern Neanderthals were living outside Africa for a long period of time. They were adapted to that environment. In particular, they were adapted to the pathogens in that environment.
Mhm.
And so we find that there is a lot of retention of Neanderthal ancestry in immune related genes. Lots of variants we have inherited from them might have helped us adapt to the environmental conditions outside. We find some differences across modern human populations too, because 50,000 years after separation of different non-African groups, there have been independent adaptations that have occurred in each of those groups. And some are also increase our risk for diseases. But it could also be that some of these variants were previously beneficial, but now with the changing cultures and diets and environments, things have become deleterious.
Yeah, that makes sense.
Yeah.
Um, so here's a question from Ralph who says, your diagrams showed a large increase in Neanderthal genome around 10,000 years ago. Does this indicate the possibility of a bottleneck in our evolution at that time? And if so, is there any speculation on what might have caused this bottleneck?
Yeah. So there are In some cases, what you find is Neanderthal ancestry or Denisovan ancestry became beneficial more recently. Now, bottleneck
Mm-hmm.
could be one reason why the frequency increased. Around that
Mm-hmm.
time that I showed you, that was about 10,000 years ago, and there were a lot of changes happening. In particular, Neolithic farming was started, and that brings about increase in population group sizes, increase in spread of diseases, new diets. And so we were really adapting to these new changes, and that might have potentially been the Neanderthal variants in those environments could have potentially been beneficial just by chance or also because of bottlenecks. And this is exactly the question that us and many others are trying to answer with looking at ancient DNA data so that we can learn about what the changes were, environmental changes were, or cultural changes were, and trying to understand how that would have led to a selection pressure to increase.
Thank you.
Yeah.
So here's a question from Ben who asks, since some people don't have any Neanderthal or Denisovan, or excuse me, or Denisovan DNA, what would their DNA look like?
Actually, so it's about one, I mean, I think our DNA would look pretty much the same. It's made of
Mm-hmm.
ACGTs.
Mm-hmm.
In some positions, Neanderthals have, say, an A allele, and modern humans have a T allele. At
Mm-hmm.
those positions, if you look at somebody who has Neanderthal ancestry, they would have that A allele. And if they have modern human ancestry, they would have a T allele. And some of these can have functional consequences, and some of them, most of them actually don't have any functional consequences.
That's very helpful, the difference between like a gene and an allele, which is a variant of the gene. So
Right,
thank you for
exactly.
explaining that.
Yeah.
So here's another question from Carlin Urate. Are Homo erectus assumed to have the same number of chromosomes as I think, modern humans and Denisovan and Neanderthals? Is there any guess when in the hominin clade history the chromosome fusion occurred?
Yeah, it's a really good question. We have no idea what Homo
Mm-hmm.
erectus DNA looks like. And so it's hard to speculate. People have tried estimating when the fusion occurred, and I think the estimates are, first of all, they're very, very rough.
Yeah.
And they are, I think, one estimate I've seen is 2.5 million years. But our understanding of our genomes, our sequencing of the genomes, all of it is getting better. And
Mhm.
so in the next few years, I'm hopeful we'll have better data that we can answer this question. I'm
Mhm.
also hopeful maybe someday we can actually have a Homo erectus genome so we can also directly answer this question. But currently, we don't know when that happened or if Erectus shared it or not.
I feel like a Homo erectus genome is in like my top 10 wish list for things in
Yeah,
human evolution that we might find. So that would be
yeah,
amazing.
absolutely. Yeah.
So another question from Carlin Urate. Svante Pabo has suggested that the genome of Neanderthals indicated some differences in neural connections than humans. Could you comment on research in that area?
Yeah, I think it's a really big question in the field of human evolution to learn about, when some of the neuronal traits that you see in modern humans arose. Did we share those with Neanderthals or not? And many people are trying to analyze this in different ways. One way, or what Swante might have been referring to here, might be to look at places where we don't have any Neanderthal ancestry, but where many modern humans share certain variants, and to try to look at those regions to learn about when these changes occurred. And for neuronal cases, I guess not many genes. So there are few genes, are about hundreds of genes, about 78 genes that have variants that are different between Neanderthals and modern humans. With the sequencing of the Denisovan genomes, we also know they didn't have these changes, but modern humans did. But the key question is now to figure out the function of these variants.
Mhm.
Um, there isn't a very clear enrichment for neuronal function, but one or two genes can also provide insights about when these changes occurred. My guess is that Neanderthals are so similar to us that many of the
Mhm.
changes that we see related to neuronal processes in modern humans are shared with them.
Mhm.
And also 60% of our genes play a role in neuronal function. And so I don't
Mm.
think there'll be a big smoking gun that, one gene separates us from
Right.
them. Although people are very actively looking into the function of each of the differences that do exist.
Yeah, I feel like also figuring out those functions is sort of a top 10 ancient DNA question in human
Right,
evolution in
exactly.
many ways.
Yeah.
So Laura asks, speaking of function, did genes or the alleles to help survive the Black Death come from Neanderthal genes or modern human genes, if you
Yeah,
happen to
I
know?
don't know if all the genes. I actually don't know what is the latest findings in this area. There are some genes. So Neanderthal genes uh, do preferentially are enriched for immune-related function. It's unlikely that Neanderthals were exposed to the same environmental pressures or, Black Death, but it could
Mm,
be that, variants that they had potentially are in the immune genes that would still have provided protection or higher sensitivity to Black Death. As far as I've looked at this literature, I don't think there is a big smoking gun that variants inherited from Neanderthals were protective or increased our risk. But just even
mhm,
extrapolating from the studies of COVID, where we saw there were variants that increased our risk for hospitalization or decreased our risk for hospitalization, and severe COVID were inherited from Neanderthals. And in both of these cases, the way I think about it is that this is just standing genetic variation. And as
mm,
environmental conditions change, something can become beneficial, or it might have been beneficial related to, say, a past pandemic.
mhm.
But same thing was also applicable again.
Yeah, that makes good sense.
Yeah.
Here's a question from Jason. Does the M2 haplogroup found in India belong to the period before the Neanderthal admixture?
I would doubt that, just because I
Mhm.
think that haplogroups are much more recent, whereas the Neanderthal mixture is much deeper in time.
Okay, thank you. I have a couple of behind the scenes questions from
Yeah.
also our team to ask you. So, one is, to what extent are we able to identify population level adaptations from ancient DNA in humans, modern humans, Neanderthals, Denisovans, or a combination of those samples?
Yeah, I think we can, as we are getting more and more data, we can actually start seeing very small changes, and we can reliably start mapping. Like, I teach selection to undergraduates at Berkeley, and one of the classical tests for looking at selection is how its frequency changes over time. And
Hmm.
for a very long time, we have not had the ability to do that very reliably because we were only looking at what exists at present. But now with ancient DNA, we can start looking at this at different time scales in evolution. And as we have more and more data, we can more reliably, really build these trajectories and then find out exactly when the frequency started increasing.
Hmm.
And so there is a lot of active work in this area going on.
That's exciting.
Yeah.
So here's another question. Could you expand more on your thoughts about the potential for finding DNA belonging to Homo erectus and Neanderthals, Denisovan, or even modern human genomes? If it's there, how might we be able to identify that that's what we're seeing?
Yeah, that is sort of, that is one of the active questions in the field. Everybody's super excited and interested in trying to see how much we can recover.
Hmm.
I think I would go back to one of the slides that I showed, which is about how modern humans are the biggest repository of archaic DNA today.
Hmm.
Because there are many, many genomes that have been sequenced of living people. Even though we are sequencing lots of ancient DNA samples, also the scale is very
Mm.
different. So we have
Yeah.
sequenced 10,000 ancient DNA samples, and we have sequenced hundreds of thousands, millions of
Mm.
living people. And so even if we inherit minuscule amounts of DNA from these groups, we might still be able to recover those footprints from our genome. And in particular, I feel for the question of Homo erectus, if it is true that Denisovans have, say, Erectus ancestry, many Oceanians and Asians have then potentially inherited some bit of that ancestry. And
Right.
so one of the things we are currently working on is trying to see if we can detect Erectus ancestry in the Denisovan bits in our genomes.
Very cool. We'll stay tuned. Maybe we'll have you back when you've
Yeah.
made a breakthrough on that. So here's another question from Ben. What's the largest amount of Neanderthal and Denisovan DNA found in a modern human?
So that question, if you look at per individual, the most highest amount of Neanderthal ancestry would be found in East Asians. They
Mm-hmm.
have about 1.5% Neanderthal ancestry compared to they have about 2% Neanderthal ancestry compared to about 1.5 that most other non-Africans have. And if you
Mm-hmm.
think of Denisovans, then that would be Oceanians who have the highest amount of Denisovan ancestry, about 3%.
Mm-hmm.
And I think there is some, because Oceanians have both Neanderthal and Denisovan ancestry, on average, they tend to have the highest amount of archaic ancestry if you combine both of them.
Mm-hmm.
And so studying their genomes can have those individuals have the highest.
Thank you. I actually now I have a good sort of a personal question. So,
Yeah.
I've had my DNA sequenced, and the company that did it gave me a percentage of Neanderthal DNA. How reliable do you think those sorts of commercial percentages are?
Yeah. So I think they are sort of reliable is the short
Hmm.
answer. So
Yeah,
one of the
fair.
reasons why they are not very reliable is that actually they are not looking at your full genome. So they
Ah.
select 1 million markers across the genome and
Hmm.
just sequence those markers just because that
Huh.
technique is much cheaper than sequencing the whole genome of an individual. Now,
Yep.
when you do that, because there is only 1 to 2% chance of having Neanderthal ancestry, what happens is you can preferentially miss a lot of Neanderthal ancestry or just by chance feel like you're detecting a lot more than
Huh.
it's there. And so they are on average correct, but they don't give you
Hmm.
any standard errors on your numbers. So you should
No.
always be skeptical about it.
Yes.
And they tend to give these, I think at least 23andMe at some point used to give t-shirts, or you could buy a t-shirt which said, I have the highest Neanderthal ancestry, or I'm in the 95th percentile. And those were wrong just because of the first issue that I mentioned, that you're just selecting a few markers and just by chance
Mm,
you might see there's more or less Neanderthal ancestry in those. But if you remember one of my slides, I showed that across within a population, most of it seems to be very similar. So there
yeah.
was
Yeah.
almost no variation if you looked at Europeans or South Asians in the amount of Neanderthal ancestry they had. It seemed
Yeah.
pretty flat because the gene flow occurred in the common ancestors.
Yeah, thank you. So we have
Yeah.
time for one or two more questions. One is another one from Carlin Urate, who asked, maybe you answered this, but is there any one particular part of the genome that is most likely to have Neanderthal genes?
Uh, Lots of places in the genome have Neanderthal ancestry. One region that seems to be enriched for Neanderthal ancestry is the MHC region. But that also has a lot of immune-related genes, so you can
Mm,
have more Neanderthal ancestry there. But in general, I think you find in many positions there is Neanderthal ancestry
sort of
present.
scattered throughout. Yeah.
It's kind of scattered across the genome.
Okay. All right. One more question from our behind the scenes team, and then we'll wrap up. So, how might proteomics or other molecular methods fill the geographic and temporal gaps in ancient DNA? And are there limitations to comparing data between these different sources?
Yeah, so I think that's a great question. And in fact, it's sort of what everyone in the field of human genetics or genomics is interested in or excited about, because there are many parts of the world where, or many timescales we have no data from, but it
Hmm.
seems proteins are preserved for longer. And so there is hope to
Hmm.
get DNA from at many time scales in Africa, or to get DNA maybe from erectus through proteomics. And one of the advantages is that we might be able to get DNA in these tough places or time scales. A disadvantage is that proteins are more conserved than genomic DNA is. And so we may not expect as many changes as we see. And so there is some concern that we might not be able to very reliably separate different groups. But for deeper time scales, there might be many differences that we might still have enough information there.
Excellent. So we can be, keep an eye on that, I think.
Absolutely. Yeah.
All right, so I'm going to wrap up. I'll conclude today's virtual program. Please join me in thanking Priya for sharing her research with us. I'd also like to give special thanks to those who made this program possible, to our behind the scenes team who helped sort through your questions, including Dr. Ryan McRae with us in the Human Origins program, to our donors, volunteers, and viewers like you, and finally to all our partners who help us reach, educate, and empower millions of people around the world today and every day. Thank you. We'll be back next month on Thursday, November 21st with Dr. Kieran McNulty from the University of Minnesota, who will be talking with us about aping human evolution, what the early Miocene tells us about human origins, kind of the other end of the timescale. We've put a link in the Q&A to where you can find information about our upcoming programs and how to sign up for the museum's weekly e-newsletter. That's the best way to stay informed on upcoming programs and to learn more about the museum's research and exhibitions. After this webinar ends, you'll see a survey pop up asking for some feedback about the program. Please take a moment to respond. We're very curious to know what topics you might be interested in seeing for future programs, and we appreciate your input. Again, thank you, Priya. Thank you to our participants, and thank you to the audience, and we'll see you next month.
Yeah, thank you for having me.